So I'm starting on this slide that I know we already did yesterday, just to remind you that in most cases when we're talking about sex-linked inheritance, we are talking about the X chromosome. Because it's the X chromosome, remember that females need to inherit 2 copies, but males only have 1 X chromosome. So if they inherit just one of whatever allele it is we're discussing, they would show that phenotype, that characteristic. So what I'd like to do is show you a cross that's not for humans, show you a cross of some other type of organism to illustrate what it looks like depending on what the male and the female parent have. So it looks like a lot of stuff. I'm going to write down what is the most important thing here so that if you haven't already printed this out, you do not have to try to recopy all of these pictures. In the first cross, we are going to do essentially the opposite of what we do in the second cross. In the first cross, parent number 1, which is the male parent, which is the female parent, has red eyes. And red eyes is what we call the wild type. It's normal. This parent, if we look at their genotype, has 2 copies of the same allele. So we would write this like this: an X with a W and an X with a W. Now this is the capital W. W means wild type. If you were just working on this yourself and trying to figure things out and you want to use a different letter, you be my guest. The actual letter that you use, unless it's specified in the question, isn't something that I would give marks or take marks away for. Parent number 2 is the male parent. So you'll start seeing fairly frequently the symbols for female and male. Female is a circle with a cross underneath it. Male is a circle with an arrow pointing up. So when I try to remember this, I always think about when I watch football with my husband, they're always like pointing at, like, I don't know, their buddy, the sky, whatever, when they score. And that's how I remember this. The male parent has white eyes. White is recessive, so I made a small w, and I made it excessively small so that we could make sure we knew the difference. Now the male only has 1 X chromosome. His other chromosome is obviously the Y chromosome. So when we take a look at which gametes the parents can make, the female can make 2 gametes, but they are both the same. She can make a gamete that has an X chromosome with a wild type allele, or she can make an X chromosome with a wild type allele. She can only make 1 type of gamete. The male could make 2 possible gametes, an X with the little w or a Y. So remember, a gamete always has haploid number of chromosomes, half the number of the parent. Since the parents have 2 sex chromosomes, the gametes will always only have 1, so that when they meet up with each other, the offspring will have 2 sex chromosomes. So when we do the cross of these 2 parents, which will be represented over here, the female only makes 1 type of gamete, an X with a wild type allele. Now, I could make a second row in my Punnett square, but I don't need to. If the parent can make 2 identical gametes, you don't have to show it twice. And we saw that yesterday in our practice question where you didn't have to use all of the squares. On the top, the male, of course, can make 2 possible gametes, X chromosome with the white eye allele, or a Y chromosome. So here is what we would see. In the F1 generation, we would see that all females had wild type eyes and all males had wild type eyes. Now the females don't have the same genotype as the female parent. The females are what we would call carriers. The females will all have one normal wild-type allele and one allele for white eyes. Since wild-type or red eyes is dominant, they have that phenotype, but now they are all carriers. All of the males we would call unaffected. They don't have the white allele and they're not carrying it. Now this is to show you that if the male parent has the characteristic, there is generally a low chance that the offspring will also show it. And that's because males only pass their X chromosomes on to their daughters, and their daughter is going to receive an X chromosome from the mother as well. In the second cross, we're going to do the opposite thing. Parent 1 and parent 2 are here. The female parent this time has 2 copies of the small w, so she has 2 copies of the white allele. So she has white eyes. The male has normal red wild-type eyes. So his genotype is that he has the normal allele on his X chromosome, and then he doesn't have another one. Of course, the gametes are when you split those 2 apart. So there are 2 possible gametes that the male can make, but only 1 possible gamete that the female can make. When I line this up and make my Punnett square, for my female, the only possible gamete she can make is an X chromosome with an allele for white eyes. The male can make 2. X chromosome with the normal allele, or Y chromosome. What will we see in the offspring? We will see all females will have red, or I'm sorry, wild-type eyes, but they will all be carriers. All males will have white eyes. So we would say they are affected. They actually have this recessive characteristic. So the moral of the story is, it is very common for a female parent to pass a recessive characteristic on to the male offspring. But the other scenario, a male parent passing it on to the female parent, is less probable. So if you see some data and it looks like way more males than females are affected by a characteristic, it probably means that it's on the X chromosome. Now, the other thing I wanted to point out is this. Females can be carriers. That means they can have the recessive allele but not show that phenotype. Males cannot be carriers. If the male has a copy of that recessive allele, that's his only copy because it's on the X chromosome and he only has 1. Males can never be carriers of sex-linked alleles. Does anyone want to ask any questions about this example? All right, the next thing that we have to go over, which is on the screen now, is a little bit about something called a bar body. This is still kind of related to X chromosomes, but now we're not talking about things being passed on. We're talking about in females who have 2 X chromosomes. A bar body is an inactive structure. And so you might wonder, why do we care about something that is inactive? And the reason that anyone would have this structure is that I'm having some trouble with words today. In any one cell, only one X chromosome is active. So This would apply particularly to females or anybody who has two X chromosomes. If you have two of them in any particular cell in the body, only one of them is actually active and doing things. It is completely random which one. So, a female has two X chromosomes. They might have the same allele on both of them, or they might have a different one, and it's total random chance which one is the one that's actually active. So I'd like to show you an example about calico cats. A calico cat is a cat that has, like the picture shows, different colors of fur, or different spots of color of fur over sort of a white base. In a calico cat, the cat would have the genotype X, B, and XO, where B stands for black and O stands for orange. Now, since there are two copies of the X chromosome, only one of them will be active in a particular cell. So if I were to look at a calico cat and I were to see the orange spot, I would say that that X chromosome was the one that was active, and the other one was inactive, so it was the bar body. If I were to look at the black spot, I would say exactly the opposite. In that particular area, B was active and O was inactive. Now, there are cases where environmental factors, for example, temperature, might influence which of the two is expressed. So, for example, in some animals, whether or not they have different colors of fur is dependent on which type of temperature they're in, which would help explain, for example, rabbits or things who change color depending on the temperature that is outside. You would not be expected to know how an environmental factor would change this, just maybe that it could. If we were talking about bar bodies, it would be unlikely that you have to do a cross with them, but more likely that you would have to explain why the cat has the different colors. Now, this might look a lot like codominance. In codominance, like in a roan horse, you see two different colors of fur sort of mixed together. And so this might look a lot like codominance. And if we didn't give you a lot of information and it was a written question, you could probably make the case for both scenarios. It could be codominance, or it could be this case of a bar body. Does anyone want to ask any questions about this example? Yes. And now in the white part of the cat, usually there is another gene completely that is also interacting with this. So that's the other thing with color, is that for some characteristics, it's not only one gene, it's one gene, and then sometimes what another gene does will affect what this gene does. So usually when you have an organism that has spots of color and a bunch of light-colored fur, there's another gene that's suppressing the production of color, and if that gene is active in another cell, then that's where that would be coming from. Does anyone else have questions they want to ask here? All right. Our next example is about multiple alleles. Multiple alleles means that for one gene, there are more than two alleles. I'm saying more than two because 2 is the typical number that we've worked with so far. Either there was a dominant or a recessive, and that was it. But for a lot of genes, there are more than 2 choices. One organism can only have 2. So there might be 15 choices for alleles. I put a picture over there of clovers. The appearance of the leaf on a clover is controlled by 1 gene, but there are a whole bunch of alleles that are possible. Each clover only has 2 of them because they only have 2 copies of each chromosome, but that doesn't mean that out in the world someone else or some other clover doesn't have different versions than that. Now sometimes when we talk about plants, it doesn't seem very real because you are not a plant. So we will talk about blood type alleles in humans. You have 3 blood type alleles. A capital I with an A, an I with a capital B, and then a lowercase I. A and B are co-dominant, and this is recessive. Now, blood types, human blood types, always shows up in multiple allele questions. I would say it is the most popular thing to ask genetics questions about because it's about humans, it's well studied, and there's lots of different things that you could ask. If you were to have blood type A, there are 2 choices for the alleles that would make blood type A. You could have 2 copies of the A, or 1 copy of A and 1 copy of the recessive allele. If you were to have type B blood, same thing, you could have 2 copies of B, or 1 copy of B and 1 copy of the recessive allele. If you have type O, which, funnily enough, is the most common blood type that people have. That is why Canadian Blood Services is always binging and saying we need O negative blood because A, most people have type O, and B, even if you don't, you can receive type O blood. To get type O, you have to have 2 copies of the recessive allele. And that's the only way that you get it. And to get type AB, which is incredibly rare compared to the others, you have to have 1 copy of the A allele and 1 copy of the B allele. Now, does anyone want to ask any questions about blood type in general before we do a practice cross? So, we are going to cross. What should we do here? This will be our male parent, and this will be our female parent, just because. Let's say that the male parent has type A blood, but the male parent has 1 copy of the A allele and 1 copy of the recessive allele. And let's say that the female parent has type B blood. Same scenario, she's heterozygous. The recessive allele is the lowercase I. That represents blood type O. Yeah, I know the letter I is there for other reasons, but 2 little i's give you blood type O. So you could say that the lowercase I is representing O, blood type O. So if you recall learning about this in Bio30E, which might be a while ago for some of you, if you have blood type A, and you look at your red blood cells, which kind of look like little donuts, they have A antigens sticking on the surface, and they have B antibodies in their plasma, so they will attack type B blood. It's the opposite for type B. They have B on their red blood cells, but A antibodies. Type O has nothing on the red blood cell. Type AB has both. So it has A and B antigens. So the type that you are refers to which things are attached to the surface of your red blood cells. Does anybody else have blood cell questions? Yeah. Could type A be? They could receive type A or they could receive type B. No, because type AB does not have antibodies. They only have the things on their red blood cells, but they don't have antibodies to attack. So we're going to do this cross. This will work like any other cross that we've done. I'll take one letter from the top and one letter from the side and match them up. Now, if I were to look at this Punnett square, what would my proportions be? I would say 1/4 or 25% of the offspring should be type AB. Another 1/4 should be type B. Another 1/4 or 25% should be A. And another 1/4 or 25% should be O. So I took two parents. One had type A, one had type B, and they could actually produce an offspring with any blood type, depending on which alleles got passed on. So I have a question for you. It's a little bit trickier than, say, the usual question. This is our expected ratios in the offspring. What if I asked you, what is the probability that their offspring has type O and that their offspring is a girl? What should I do? So I'll back up. What is the probability that it would be a girl? If two people had a baby, what are the chances of it being a girl? 50%. You got it. So there is a 50% or 1 out of 2 chance that is a girl. What are the chances that she has type O blood? 25%. Does anybody who has taken or is in math know how to combine probabilities? 50% times 25%. So 50% is a half. 25% is a quarter. That gives me 1 out of 8. It would be likely for at least one question on a test to ask you both of these things. Ask you to do a cross, you'd get a percentage, and then the question would ask you, now what's the chances of that and it being a girl or and it being a boy? So you could multiply fractions. You could do 0.5 times 0.25. You could do 50% times 25%. Whatever method you prefer using in your calculator is fine. But if we were to ask you about a characteristic that's not related to the X and Y chromosome, and whether or not it was a girl or a boy, this is what you would do. Does anyone have questions they want to ask about that? So totes going to be a question on your test next week that asks you this. All right, the next thing that is possible with multiple alleles is something called the order of dominance. So we're still talking about multiple alleles. With blood type, 2 of them were codominant and the other one was recessive, but that's not always the case. In cute little rabbit fur color, there are 4 different alleles: agouti, chinchilla, Himalayan, and albino. And they have an order of dominance. This is the dominant allele. It's a capital letter, so it is dominant to everything else. For the other ones, they all have lowercase letters, which means they are recessive to that first one, but not necessarily to each other. According to this, chinchilla is recessive towards agouti, but dominant towards Himalayan. The same thing is true for Himalayan. It would be recessive compared to chinchilla, but dominant compared to albino. Now, albino is the last one. Albino would be recessive to all other alleles. You would not be expected to have known this. We would always give you this information or tell you what was dominant so that you knew what kind of combinations you could make. Here are the possibilities for phenotypes. So what do they look like? And genotypes. What possible combination of genes could give you that phenotype? So for agouti, recognize it doesn't matter what the other letter is. As long as there is one capital C, then that is the phenotype that's going to result. There are 4 possibilities. Agouti with any of the other alleles, regardless of which one it is, will give that phenotype. Chinchilla is the second one, and if you have the printed notes, we have to add one. But for chinchilla, it will be the phenotype as long as agouti is not present. If it's there with any of the other alleles, so 2 chinchillas, chinchilla and Himalayan, or chinchilla and albino, for any of those, chinchilla is what you get. There are 3. For Himalayan, there are 2. Either 2 copies of the Himalayan allele, or Himalayan plus albino, because that's the only one it's dominant towards. And for albinos, there is only one possibility. If it's the most recessive characteristic, you have to get 2 copies of that allele. Now, in the practice questions that we'll work on tomorrow, we will do a bunch of examples with this scenario. So we'll just do one quick one right now, just to give you a bit of practice. Let's say that I take this cross. So I'm going to cross a Himalayan. I know it's Himalayan because I have an H and a C, and that was one of the choices for Himalayan. And I'm going to cross it with a chinchilla, this particular genotype. So what I would like to know, and I want you to just take a couple minutes here, I would like to know what are the phenotypes that are possible for the offspring? What colors are possible? So hopefully you were able to fill in the square, but perhaps figuring out the phenotypes is the more challenging part. Since you have the list up here of which genotypes give which phenotype, we'll just match them up. This organism here has a CH and an H. That matches up with chinchilla. The next organism on the top row has a CH and a C. That also matches up with chinchilla. So I'm going to have 2 out of 4 chinchilla. The bottom 2 both represent Himalayan. 2 CHs or a CH with just a C. Here's 2 CHs, there's CH with just a C. So 2 out of the 4 would be Himalayan. The idea is that you cannot produce an organism that's more dominant than any of the ones that you had. So since I started with chinchilla and Himalayan, I might have been able to produce an albino depending on the genotype, but I should not be able to get an agouti rabbit because that dominant allele was not present. If it were, I would have seen it. Does anyone want to ask any questions about this cross? So the next one. The next one is about polygenic inheritance. Polygenic, many genes. Polygenic inheritance is when many genes, in other words, more than one gene, control a trait. So a really good example of something that is a polygenic trait or that is inherited polygenically is something like height or something like color. Skin color is the example that I have here, but it could be hair color, it could be eye color. All of those things are polygenic traits. In my little example here, I'm just using it to represent how complex this can be. I will never ask you, never, to make a 64 square Punnett square and fill it in. That would be silly and ridiculous and take up way too much space and time. What I want to represent is this. Let's say these are my two parents that I'm crossing up here. Both of these parents have medium colored skin. So neither of them has particularly dark or particularly light colored skin. But depending on their genotype, for 3 different genes, A, B, and C, there are a lot of possibilities for their offspring. These two parents with medium colored skin could, it's a 1 in 64 chance, but could produce an offspring with very, very light colored skin. So someone who was close to being albino, we would say. They could also produce, albeit by a small chance, someone who has very, very dark colored skin. And that's because there are many genes involved, and the combination of all 3 of them is what determines what skin color you have. So the idea is this. If you have offspring that do not seem to resemble either one of the parents, which is how I often feel in my family. If I look at my parents, I'm like, I don't belong to either of you. I have to remember that this comes into play for a lot of human characteristics, like height, like the color of skin and hair and eyes. So if you are looking at your family while you're asking them all these questions about your family tree assignment, and you start wondering, boy, Uncle Tom doesn't really look like anybody in the family. I wonder where the heck he came from. This is probably what went on. Generally, we'll see the proportion look sort of like this, where most of the offspring will have what we call just the medium, the average, not the darkest, not the lightest, or not the tallest, not the shortest, but that it's possible to have offspring with the extreme characteristics. Oh, we'll talk about height for a second. Wouldn't it be funny if everybody in the world was either tall or short, and that was the only 2 choices? It would be weird. There would be a bunch of tall people and a bunch of short people. But it's not. If you look, I'm sure, at your own family, I think it would be unusual for everybody in your family to be exactly the same height. So one of my brothers is 6'4". My grandmother is 4'11.75". She likes for me to say, because it's almost 5 feet tall. But that's a huge range of height. And we're all coming from the same genetic background. So what would you have to do if there was more than one gene? You might have to do this. Complementary interactions. This is the type of question that might get asked. Rooster combs is the classic example of complementary interaction. Over here, I put some pictures of what different rooster combs, so that's like the feathers on the top of their head, might look like. The R allele, capital R allele, is dominant, and it produces a rose comb. So this one over here, just so you have a visual, is what the rose comb looks like. I don't know who made up the names. It wasn't me. They're kind of silly. There is another allele, P. R and P are different genes. So they are not the same gene. We have two totally separate genes that are involved in the shape of the comb. The P allele is dominant, and it produces a P comb, which is that one. Now, if by some chance an organism has both dominant alleles, so they have at least one capital P and at least one capital R, I'm putting a question mark for the second allele for each of them because I don't know what it is. It could be a uppercase or a lowercase letter. But if they have at least one copy of the dominant allele for both of them, you get a different looking comb. So if I only have the dominant rose allele, it's that shape. If I only have the dominant P allele, it's that. But if I have both of them, you get the walnut-shaped allele. Here's my paper here. Walnut. So if you have both, you get walnut, which is not P and it's not rose, and it's definitely not a mixture between the two. It's totally different from the two that we started with. If you have neither, so if the organism is homozygous recessive for both of them, then you get a single or simple comb. Now, those are the types that we're going to focus on. Obviously, they are not the only ones that are possible, but those are the few that we're going to talk about in this particular example. We are going to start out by crossing these two organisms. They will be our parents. Then we will cross the F1 generation. In my parent generation, we'll start with, we'll call that female, we'll call that male. Female, male. Now, what kind of gametes can my female make? Gametes are haploid. They have half the chromosomes that the parent had, but they have to have one of each letter. So I could not make a gamete that was capital R, capital R. That's not half the chromosomes. I've totally taken one away. I could, however, make one that was capital R, lowercase p. That would work. Now, for the female parent, do I have other options? What other gametes could I make? The answer is none. That's the only gamete that she makes. My other choices are capital R with a P, capital R with a P, or capital R with a lowercase p. Now, like I showed you yesterday, you can do that. Your answer won't be wrong, but you will have done a ton of work that you don't need to do. So I'm not going to. If you have all identical alleles, please don't repeat them because you're just going to end up doing extra work. The male parent is the same scenario. He can only produce a gamete that has a small r, because that's all he has, with a uppercase P. That means that I don't need anything in my Punnett square except for the very first box. All of the offspring will be heterozygous for both characteristics. So, in this cross, I took a rose, combed parent, and a P, combed parent. All of their offspring are walnut. So none of the offspring would have the same phenotype as the parents. In the F1 generation, or the F1 cross, is where we're going to take two of the descendants, so two of the F1 generation, and we are going to use them down here to make the F2 generation. So I would like for you to try this cross, so that you know you should have more than one gamete for both of them. So the size of the Punnett square is more representative of what you will need to use for this second cross.